Welcome to the 45th webinar of the FCTL webinar series. So this week only we have completed around 2 years sharing the knowledge in different fields of the food science, technology, engineering and related areas. So this is our 45th lecture and it's all about sharing idea, knowledge and from the alumni of FCTL IIT Kharagpur to the young minds or the working professionals in the field of food science and related fields. So today I would like to welcome our young and dynamic researcher, that is the Dr. Bhanu Devnani, who is currently doing her postdoctoral from University of Massachusetts, USA. And before going to the formal introduction, I would like Mishra sir to say some few words about her.
So good evening.
Thank you, Subham. So
Yes.
I think people are more eager to listen to Bhanu. But anyway, Bhanu has been one of the best MTech student of our department, and she was not only MTech but also she is the best all-rounder. When she was there for 2 years during her MTech program, she was also involved in so many extracurricular activities, and which definitely helped her. After I think she went in the campus only for 1 year because her MTech project she did under DART. She went for the DART project in Germany, and that project again further she continued later as a PhD. She went, of course, after completing the MTech work, she came back to India, submitted her MTech thesis, and then within a few months, she again flew to Germany. I think while she was doing MTech, only you applied for your.
Yes, sir. So initially I applied through Melbourne, India postgraduate program.
MIPP postdoctoral program, and that later it was converted as a full student.
Melbourne Research Scholarship. Yeah.
So yes, and she has been a very bright student, and which is already indicated from her career graph. After doing PhD, just now we were discussing. Recently, she has gone back to US now for doing postdoctoral. Yes, welcome Bhanu again to your welcome, and we are excited to hear you. Let's over to you, Subham.
Yes. So like this. मैम सर ने तो बोल दिया जो था, अब हम थोड़ा वो दे देते हैं, फॉर्मली इंट्रोड्यूस कर देते हैं आपको मैम सबसे.
बिल्कुल.
She was senior to you, Subham. How many years senior to you?
2 years.
2 years.
Yes sir. चिरस्मिता मैम के बैच के थे ना मैम आप?
अच्छा, चिरस्मिता के बैच के.
फेलोशिप का पूछने के टाइम पे बात हुई थी मैम से पहले एक बार.
चिरस्मिता तो है. चिरस्मिता, वीडियो ऑन करो. Then you should have invited Chirasmita to tell about. तो जो चीज हम नहीं जानते वो बताई होती, चिरस्मिता बताती. ठीक है,
चिरस्मिता मैम भी होगी यहाँ पे अभी. ठीक है. यस सर.
चलो, go ahead.
Now, coming to the formal introduction. So, as we all know that we are currently inviting Dr. Bhanu Devnani. So, she has currently working as a postdoctoral fellow in University of Massachusetts. She has completed her BE in food technology from Punjab University, Chandigarh, and MTech in food process engineering from IIT Kharagpur. And after that, PhD from the University of Melbourne. So during her MTech also, she got that fellowship for completing her project work from German Institute, where she worked in Karlsruhe Institute of Technology, Germany, and her whole topic of area was wholly related to the plant and dairy-based meat alternatives or dairy alternatives. So in her MTech also, she worked on the development of meat analog using high moisture extrusion processing. During her PhD tenure, Dr. Devnani worked on the study of behavior of almond proteins and purified minimally processed and complex food systems. Along with research, she was the graduate research project mentor and helped in designing and mentorship of 3 research projects related to almond protein. Later, she joined as a research assistant in University of Melbourne and currently now working as a postdoctoral in University of Massachusetts. So she has a technical expertise on protein structural analysis, protein and product stability, protein composition, microscopy, and all the related analysis related to the protein. She has attended many international and national conferences and published peer-reviewed journal papers in the journals. So now I welcome Dr. Bhanu Devnani to kindly deliver her seminar on the topic structuring of plant-based dairy alternatives and their challenges and opportunities. So kindly all the participants mute their mic, and if they have any queries, so then put it in the comment box. It will be discussed after the seminar or the webinar. Thank you. Bhanu ma'am.
Okay.
Bhanu ma'am, you can share your screen.
Okay, that's weird. I just shared this window. Apparently, it doesn't seem to. Probably I'll just share the entire screen.
Yes, ma'am. You have to share the entire screen. Otherwise, it will not show in the presentation mode.
Oh, okay, okay.
Entire screen, and then you click, then share button will activate. Entire screen पर click करो, and then नीचे share button will activate. Share.
Sir, when I'm going to entire screen and तो a pop-up is coming up and उसमें जो share है वो grey आ रहा है.
शेयर ग्रीन नहीं आ रहा, उसको तो क्लिक करो, पहले इंटायर स्क्रीन को क्लिक करो, उसके बाद फिर शेयर बटन एक्टिवेट होगा, और फिर उसको क्लिक करो.
अच्छा, हाँ अभी हुआ, right. So you all can see my screen?
Yes.
Yes, ma'am.
Okay, perfect.
उसको हाइड कर दो, एक ओपन डायलॉग बॉक्स ओपन है नीचे, उसको हाइड कर दो. यस.
Okay.
That's fine. Now it's fine.
Okay, thank you so much. Thank you so much, Shubham, for such a nice introduction, and thank you so much, Mishra sir, for giving me this opportunity to share my knowledge on whatever like I have done until now. Almost like the entire thing I'll be like touching base on. So today I'll be talking to you guys about structuring plant-based dairy alternatives and what are the challenges, and from there, of course, the opportunities come up. So a little bit of disclaimer for the camera, because I am available on camera from my phone and I'm presenting on my laptop, so my face might be like here, there, everywhere. So I apologize for that. For some reason, my camera in my laptop is not working today. So apologies for that. And also, I'll try to keep this webinar interactive. So please participate and make it more interesting. Okay, so let's get started. So first of all, when we think about plant-based dairy alternatives, this question of why plant-based is not a question anymore. Everybody almost knows why plant-based. So, back in 2016, when I was starting my PhD, or actually in 2017. So 2016, I started my research, and it was on plant-based meat alternatives back in Karlsruhe, Germany. So plant-based meat alternatives, especially from soy, had been there for a while. Also, soy milk had been there for a while, but not very popular in India because we all are dairy lovers in India. So but back in 2017, this field was at a very, very infant stage. And, it was... I remember back in 2016, in the US specifically, almond milk became like a really big thing. It was like the toughest competitor of soy. And, so that actually raised the market, or I can say like it basically broadened the market. Like after soy milk came almond milk, and then, now like we have plant-based milk alternatives from like a variety of plant-based sources. You name it and you can milk it. It's like that. So why plant-based? Very commonly known now. I'm so glad that this field is also becoming very, very popular in India now. So there are like a lot of vegan chains like Good Dot good milk was developed by Abhirangan in like 2019. So he was the main person who actually started the plant-based milk idea in India, and I'm so glad to see that it's like the people are responding to this idea really well. So why plant-based? So be it lactose intolerance or milk allergies, or people consider it to be healthier, or to reduce the environmental impact that dairy has on the environment. So for like a couple of reasons, people want to shift to plant-based diets. Some people, they are flexitarian, so they are happy to have like the regular meat and dairy, and they also want to do incorporate plant-based stuff because they consider it's more healthy. So when we think about plant-based dairy alternatives, it is not just milk. It is the millions of products that we get from milk. So it's yogurt, it's cheese, it's butter, it's ice cream, a lot of products. So for today in this webinar though, I'll be focusing on the products which are mostly gelled products. That is like when we form a gel during their processing. So, and why that? Because that gel formation is actually directly linked to the protein properties, which is the focus of my area. So I will be focusing more on like the yogurt and cheese-like products, which involve that gel formation and involve quite a lot of protein network and cross-linking. So when we think about plant-based dairy alternatives, like we can follow two approaches. The first approach can be like minimally processed systems, when we have like for example, you take almond, you just soak it in water. Next day you grind it and strain it to obtain almond milk. Minimally processed. And the other way is that when you use purified systems, you take protein isolates, you mix it with a certain amount of fat, and you mix it with certain amount of like gums and stabilizers, etc., to get a particular product. Now, both the ways have their own advantages and disadvantages. For instance, when you look at minimally processed systems, I particularly like that because it definitely retains the source nutrients. So their nutrition profile is much more diverse as compared to when we make anything from purified systems. Also, it is very easy to get a clean label product in that case, which is loved by the consumers and also loved by the environment, because we are actually putting much less energy to make the product. So, but then, the use becomes a little bit limited. We can use it in, plant-based emulsions, or we can use it in, the gels that are formed from plant-based emulsions. But if you think of a broader market and the broader product space, then this purified system is actually really good, because, you can use protein isolates like in a lot many things. So you can use it in snack bars, and you don't even need to just restrict to daily alternatives. You can use it in sports nutrition, snack bars, etc., etc. So there are like a lot of advantages that you get. This particular advantage, that you get greater flexibility in formulations, and also you can easily get like high protein content. You can tailor, how much protein do I want to put into the product. So that is really good in terms of using purified systems. So now that we know that like there are two ways of making it, so I'll share which particular ingredient did I use for my research, because the thing is, there are so many plant species that you can actually milk any of those, whatever you want to. So we have to choose something. So we definitely chose the almond, the mighty almond, I would like to say. And why I say that, because it's pretty good. So I don't need to like say in India, we already know, badam khao to usse matlab acha brain chalega. It's like our daddies and nannies have always been saying that to us. So obviously it's nutritionally very, very rich. And the second thing was that it was local to us in Australia. Specifically, I was in Melbourne. So Melbourne falls in the state of Victoria, and Victoria alone is responsible for like around 66% of the almond production. So because it was like local to us, so we thought it's a very good idea to strategize and use something which is local to us. So again, with the whole idea of like spending less energy, something that's local to us is better for us and for the environment. Also, as I mentioned previously, that almond milk, this is like back in 2016, 2017, it was giving a tough competition to soy, and then slowly steadily it also like crossed the sales of soy. So the popularity that almond milk had gained at that time. So all of these reasons, the nutrition, its local availability, and also the market size actually made us feel that this can be a very, very good raw material for our research. Acha. We selected the raw material. Okay, almond is fine. So what did we do next is that we thought like, okay, there are a few almond-based products already available in the market. So let's just have a look. How are they? Where is the gap? Like, where is the opportunity? और क्यों है वो मेन चीज ये फाइंड आउट करना इंपॉर्टेंट है तभी हम किसी चीज को इम्प्रूव कर सकते हैं राइट. So what I did was I took like 3 almond-based yogurts, which were like the only almond-based yogurts that were available in Australian market at that time. And I also took like a soy yogurt and dairy yogurt. So here AY1, 2 and 3 are basically representing all almond yogurts, the different formulations that I took. And soy and SY and DY represent soy and dairy yogurt. So like the first and foremost thing to understand or to know ki what is happening with the product, like chal kya raha hai kya cheez pehle dekhein. So I love this thing about microscopy. You can get to know like a lot of things, and it's just so beautiful, like beautiful images telling you a lot of information. So I really like microscopy. So as you can see here, अगर हम soy and dairy yogurt में देखें, so we get a continuous protein network there, which is not there in any of the almond yogurts. So again, fat is red here, protein is green, and starch is blue. So we can clearly see that there is no protein network in any of the almond yogurts. The second thing that we can see is that the particle size in almond yogurt is pretty big as compared to soy and dairy.
যদি আমাদের একটু স্লো কাজ করে একসাথে করলে 1 ইঞ্চি লিকুইড হয় ওটা ঠিক আছে ওই জন্য আমরা কি করছি জানিস তো এজন্য আমরা দুজন লোকের থাকলেও কি বলতে একটা উপর
তোমার কাইন্ডলি মিউট ইউরসেলফ
Okay. So sorry about that. So we can see 2 major differences: the protein network, which was absent in almond yogurts, and also we can see that the particle size in almond yogurts is very big. Now, when you go into the literature and you see, chalo ye to microscopy ho gaya, right? But then what is the impact on the product? And how do we get to know that? So we have certain rheological measurements. One, the most commonly used for yogurt is viscosity. So if you go into the literature, you'll read like a lot of articles saying that viscosity is directly proportional to thickness, it's proportional to creaminess. Now, so that's why we measured viscosity first of all in the first place. So as you can see here, the viscosity of like almost all the yogurts was similar. So if you include the error bars, we did not detect any significant difference in here. So the difference was just not there. So that actually made us feel that the yogurts were designed by the industry to have a similar viscosity. So now I would like to ask you that what do you think? The viscosity is similar, we know that. The literature says that viscosity is proportional to creaminess, but looking at the microstructure and the particle size differences, what do you feel like? Will the smoothness or the creaminess of almond yogurts be similar to that of soy or dairy? Which one would be higher, lower? What do you think? I want to know.
Yes, students in the group, please respond. Those who are, I find many MTech students are there. Please respond. Raise your hand and respond. I think you proceed, Bhanu. They will not.
Okay.
And then actually the soya DY has the highest creaminess, I think, because of its protein structure. The very small particle size is there, so it will be more uniform and more creamy as compared to almond.
Okay. Okay, so let's see what did I get. So this is basically a graph showing the tribology measurement. So tribology basically measures, it's an instrumental measurement, and it actually measures how creamy or how smooth we can feel the product. So as you can see on the y-axis, we have friction. So the more the friction, the lesser the smoothness is. So you can see here that as anticipated, the almond yogurts, they have a bigger particle size, so that's why they have like a higher friction coefficient and therefore lower smoothness. A by 1 and A by 2. A by 3, although is like a bit different, and dairy is kind of neutral. So I would like to I would like you to focus on the entrainment speeds of like 10 to 100 that I have actually have a box on it here. So the reason why I have put a box on it, because this speed 10 to 100 mm per second is actually the speed that we experience most when we chew or when we eat anything. So here you can see that A by 1 and A by 2, they have higher friction, and which we anticipate is because of the particle size. And the thing is, with dairy, it's just straight. It's like this, the friction coefficient is just remaining the same. Whatever is at the start is almost the same at the end. Whereas with the plant-based, you can actually see that with increase in speed, we are also having an increase in friction. Now, A by 3 here is it's a bit different formulation, and the smoothness is rather higher, the friction is lower, and the reason for that is because the A by 3, I would say it was a very intelligent formulation. It had coconut fat in it to give that melting feel or mouth feel, but the thing is, it is still different to dairy, and the reason is because the melting temperatures of coconut fat and of dairy fat are different. Coconut fat melts at temperatures like around 24, whereas dairy fat melts around the body temperature, so they do give different feelings, mouth feels. But yes, definitely there are like a lot of differences in here. So now we know that like the viscosity was similar, right? But the friction coefficient is giving us a different story altogether. The products are not similar, whereas the industry just looks at the viscosity and they're like, oh, but your product are good, Lekin, if viscosity is not governing friction coefficient, what is it? What is governing the friction coefficient? So basically, the gel strength was governing the friction coefficient. Again, the blue box that I'm showing here is showing you that how the products, they are changing with increase in frequency. So here on y-axis, I have tan delta. So this graph basically gives us the idea that when we increase the shear, when we increase the frequency, how are the samples responding? So if our G double prime increases, that means the samples are becoming more liquid-like with increasing shear. So again, here you can see that the dairy is consistent, but all the plant-based yogurts, they are increasing in their liquid-like behavior, or their shear thinning, so the blue boxes in both these figures of the friction coefficient and the tan delta, they correspond to the same speeds. And if you see closely, the graphs will be, they are in the same correspondence, but in an opposite way. So basically, the friction coefficient or the mouthfeel was more governed by the gel properties rather than, just the viscosity. Okay, so after knowing that, the question that came up was, now we know this, just long story short to summarize it, that dairy gels, they have an interconnected protein network structure, and the protein and fat are governing the texture and consistency. Whereas in a non-dairy gel, we know that it's basically a starch and gum-based matrix, and there are clusters of protein and fat coming together, which are basically responsible for texture and consistency. So we know that this is the base difference, and because the base is different, the properties are definitely going to differ. Specifically, if I talk about the gel properties, what should we do? So I discussed about two approaches earlier, using a minimally processed system and using a purified system. So I took both the approaches. So first, focusing on the left, I took a minimally processed system, that is almond milk. Just take almonds, milk them in a normal way, and then heat and pH are the two most common variables that are used to modify the gel properties. If you look about making yogurt, the way in India we make yogurt, we heat the milk a bit, then inoculate it, right? So heat is a very common variable which is used to change the protein properties, and obviously pH alteration as well. So when I started at that time, there were a lot of gaps in literature because we didn't know, like, I didn't know the answer to what temperature do I want to heat my almond milk to if I want to, use it for yogurt purposes or other purposes. What do I want to heat it to? What pH do I like? How do I change the pH? And how, if the pH changes, would the gel properties change? So there were like a lot of gaps in there. So to answer those questions, we took both the systems, minimally processed and purified system as well, and we studied the impact of heat and pH on these systems. So now if you look at the microstructure of the minimally processed almond milk, so you will see how the fat globules are there and they are surrounded by the protein membrane. Now, this microstructure of almond milk is totally different to the purified system that you can see on the right-hand side, which is of almond protein isolate. The proteins, their sphericity, their size, and other properties, the extent of denaturation also was massively different. So you can see that, like on the left side, in almond milk, we were getting the particles, like bigger particles, and so we had like protein clusters which were like around 400 nanometers, and then obviously fat particles were bigger, of course. But if I talk about only the protein and purified system, which was like 90% protein isolate, the particle size was much smaller, which was like around 12 nanometer. So you can see how different the particle size is as a result of the raw material processing. One is 400 nanometer, one is 12 nanometers. So we are starting with, even though it is from the same raw material, it's from same almonds, but still we are starting with very different systems, two very different systems. So let's see what we saw. First, I'll be taking you through the almond milk system. Very, very simple experiment. You take almond milk, you heat it, and you see what are the changes that are occurring. So I also skimmed off, I made skim almond milk from there, took the fat out, just centrifuged it and took the fat out, and then concentrated the protein in the skim almond milk through centrifugal filtration to do some, tests on how the protein changes. So to focus more on the fundamental aspects, so we did hydrophobicity index measurement using the ANS reagent. And as you can see here, so we heated in a broad range of temperatures, like from around 45 to 105, and that range was selected on the basis of literature. So as you can see here, we got a massive increase from 65 to 75 degrees Celsius. Something is definitely happening in there in the milk. Protein is definitely changing. Looking at the CD structure, we saw that at 75 degrees Celsius, not at 65, but at 75, we could observe changes in the secondary structure as well. Going further, when we did the SDS phage, so what we did is we calculated the densitometry of the SDS phage gels through ImageJ, and this is what is being shown on this graph. So we could see the band intensity, or in simple words, if I say the thickness of the band, changed with increase in temperatures, because if more protein is denatured, it will settle down. The supernatant will have less protein, and you will get a fall in the band intensity. So from 75 to 85 degrees Celsius, we saw that drop. Okay, we definitely know something is happening from 65 to 85, but what is happening? Again, let's put the sample under the microscope. So at 20 degrees Celsius, as I mentioned, the proteins were in form of clusters, and the size of these clusters was around 400 nanometers. And when we heated at 55 degrees Celsius, so 45 we started, we did not see any change. But at 55, this was very interesting to see actually, that the clusters are actually coming together. They're maintaining their spherical shapes, but they are just coming together. Increasing the temperatures to 65, we could actually see that they are not spheres anymore. They have changed the structure, they have opened up. To increase the temperature further, we could see that these opened up structures are coming together and aggregating together. And finally, at 85 degrees Celsius and 95 degrees Celsius, of course, as well, we got a weakly flocculated gels. Now, these images that I'm showing you have just green in them because it is all protein. These tests that I'm showing you, they are all for just skim milk. and that is why they're all green. So the similar tests were also carried out with a full fat milk, and you can definitely check out the full article, which I'll link here as well. So here, because in the interest of time, I'm just showing you the results for the skim milk. And to like further confirm like the gel point, we also gel the milk in the rheometer to see like what is the gelation temperature TG that we are getting, and if we increase the protein concentration, does the TG decrease? So we found that the almond milk in particular, it will gel just through heat at a temperature of 88 degrees Celsius, and when we increase the protein content to around 11%, so the gelation temperature reduced to around 80 to 83 degrees Celsius. If you want to have definitely a further look, you can definitely check out this article that I'm linking here. Now, this system was now totally different to the next system that we took, which was basically almond protein isolate. Abhi protein isolate ke liye what is the most important thing? For it to be useful, it needs to be soluble, right? So the first thing that we checked was solubility in the case of protein isolate. So the interesting thing was that the protein isolate was very well soluble at like pH 4 and pH 7 as well. So the solubility was pretty similar. And with proteins, like we usually struggle with this, that at lower pH, they are not really soluble, and, their applications become limited, which was not the case with almond protein isolate here, which was good. So they were soluble, which gave us like a heads up. Okay, we can go and use these in beverages as well. So pretty broad application. From beverages to, of course, I said other applications like snack bars or whatever. But for beverages, we definitely need to have solubility. Now, the interesting bit is their solubility is similar. You run them through SDS page and you actually see that there is difference in the protein profile. Now, what then, we got into some further analysis using LCMS, and we actually saw that the big band at pH 7 that you are able to see, which is around at 62 kilodaltons, that is the major almond protein, amandin, which makes around 65% of the total almond protein. So it actually broke down into smaller molecular weight proteins at pH 4. So we were happening at pH 4. And that then influenced the protein functionality. So we measured the stability through DSC, and we did not find any peak at pH 4, whereas pH 7 denatured at like around 82 to 88 degrees Celsius, somewhere there in that range, similar to what we found in the almond milk as well. But at pH 4, we also got enhanced foaming, probably because the particles or so the proteins had, hydrolyzed into like smaller molecular weight proteins, so they were more flexible to orient themselves around the air, like the membrane, or you can say around the air globule. Also, another thing is that when we did CD, we did see that pH 4 samples or protein was actually a lot denatured, which obviously also helps the foaming capacity. Now, very interesting result here, very different from like what I have read in standard papers. Very, very different results. It took me quite a bit of time to actually understand what is actually happening with the system. So we got concentration-dependent age thickening at pH 4, which is very interesting, and it was a very new concept to me that aisa bhi hota hai kya? My reaction was that. So with time, the viscosity of the solution is changing for different concentrations. So at 3%, with time, you won't get any change, which is okay for the concentration of beverages, right? But if I'm trying to use my almond protein, I'm trying to sell the almond protein powder, and so there I have like people are using bigger concentrations, like you easily use anywhere between 6 to 9% of concentration. What's happening? When you do the concentration dependent, when you see the viscosity, you can actually see that the viscosity is increasing with increase in storage time. And this is what was also happening at 9% concentration. So the more you increase the concentration, the quicker the solution thickness will increase. Okay. The question of like why this was happening, again, we dig into LCMS and thinking like, okay, is enzymatic hydrolysis triggering these things? What is happening with the systems? But actually we could not reach to a certain conclusion, of like why this particular thing was happening and what was leading to this. Coming back to microscopy, and as you can see the arrow, that at 9% concentration, we already had like a jelly-like gel, probably I can say. So, and the microstructure was again very different to like at pH 7, it was still a liquid 9% protein solution, whereas at pH 4, we already had a gel. And when we heated these gels, again, we could see like a cross-linked network formation at pH 7, which is again, if you correlate, will be similar to that of the almond milk. But at pH 4, the gel was totally different. So we could get heat-induced gels with different structures and molecular interactions by tailoring the heat and the pH. Now, if I compare this system with the system before that I showed you, so if I compare the milk and the isolate, what we actually found was that moving from minimally processed to purified systems near pH 7, they altered the protein size, as I already discussed with you. Massively different, altered the sphericity and the degree of denaturation. And as a result, when we measured or when we carried out rheological measurements on these gels, we saw that the gels that are formed with the protein isolate, they're much finer and weaker as compared to the gels that were formed with almond milk. So in terms of gelation, basically, almond milk is better as a raw material for me as compared to if I use a highly processed protein isolate. So yeah, this is what I wanted to share with you, and I would definitely like to thank my supervisors. I forgot to actually mention in the start. So my current affiliation is with the University of Massachusetts, but this position is relatively very new. It's just been 2 weeks here for me that I have joined. So this work that I'm presenting is all my PhD work that I carried out at University of Melbourne. So would definitely like to thank my supervisors, very supportive, very encouraging, and very lovely people. So they were all like very, very supportive and encouraging throughout my entire journey. So a big heartfelt gratitude to them. And thank you so much for all of you for your time and for listening to me and for this and being there in this webinar. Thank you so much.
I think, Shubham, there might be some questions.
A very informative and nice and crisp results you have shown. Like you have,
thank you.
Like ma'am, you have shown the gist of the whole PhD in just 4-5 slides.
Yes. Yes.
It's nice presentation. Well done.
Thank you, sir. Thank you.
Yeah. I think questions.
If any participant has any query, you can ask.
You can write in the chat box or you can directly ask.
Yes, sir. Good evening, sir. I'm Gargi Hushal. Am I audible?
Yes, you are audible.
First of all, thank you so much, sir, for organizing this type of webinar. And I congratulate Dr. Bhanu. She is my student also in Punjab University, Chandigarh. Congratulations, Bhanu. Nice presentation. Yes, sir.
I think you are from here, IIT?
I'm from Punjab University, Chandigarh. She's my student in undergraduate course.
Okay, great.
Oh, yeah. Congratulations, Bhanu. Thank you for very good presentation. I just want to ask one question. What about the difference between your cow milk yogurt and your almond milk yogurt and basic shelf life and all? You didn't mention. So, what about the shelf life and stability and all?
Okay.
Yes.
So, in terms of actually stability, thinking about that. So first of all, because the almond protein is very, very different to dairy protein. So if we use the same conventional method to make, almond yogurt compared to dairy yogurt, we won't get a set gel in that. So that is why, like most of market products, they always have like Bunsen stabilizers added into it. And one thing that I don't like is that most of the industrial products will have a very low concentration of protein, which I can understand is because almonds are expensive, and if they increase the almond content, if they increase the protein content, so all the results that I mentioned here, basically, so we worked at like particularly high concentrations, and my starting concentration was like at least 3%, 3.5% to see, you know, if it is comparable with dairy milk. But still, if I use that concentration, it won't set as a dairy yogurt because the protein properties are very different. So it won't set. So that is why to enhance the stability, we always have to add. So the market yogurts that I've assessed, basically they were all less stable. We ran them through Lumifuge and they just collapsed within the first minute. So they exhibited a lot of synergies, even though they had gums and stabilizers, but because dairy had the set gel, it did not collapse. So we definitely have those differences based on like, one sets, one doesn't set. So one is more stable, which sets, and which doesn't set, it's not stable. So we had those differences.
And this kind of any product is available in the market commercially?
I'm not sure about India, ma'am, but I'm definitely, so here in the West and back in Australia, so there were a lot of products, but I'm not sure if India has almond yogurt yet.
Okay, Bhanu, thank you so much. Very nice presentation. Once again, congratulations.
Thank you.
And thank you, Professor Mishra also. Thank you, sir.
Okay, I'll try to look at the messages in the chat box. Can I stop sharing my
screen? I will read it for you. No problem.
Hello.
Yeah.
Manu, I have a question that is there any effect of education on stability of the yogurt while mixing the ingredients? Have you?
Oh no,
is that
any microbial analysis? So we didn't do any microbial analysis, so I don't have information on the sterility of the yogurt.
No, sterility. Sterility meaning stirring, agitation.
Okay. So yeah, again, we didn't actually like stir the yogurt and see like how it would, or stir the gel as such and see how it would behave. But based on what I have experienced, I can definitely say that there might be water separation, and which is why I think majority of the market products have stabilizers added to them, like these kind of products, the almond-based ones.
Okay, thank you.
And there's a question from A.K. Sharma, like, what is your opinion on peanut milk and its product?
Oh, well, peanut is a very good source, and I would say it's much cheaper than almond, so it's really good that way. And I have actually read several articles on peanut milk. In fact, like in India, peanut yogurt is being sold in the market. So when I was back in India for like a few months, I actually explored the products that are there, the vegan products. So the good part about peanut is that it actually sets, to say almond ke saath, there is this challenge that it won't set, but with peanut yogurt, it actually sets like a proper dahi, so which is a very good thing. But the problem in US and Australia, like Western countries, I would say what I've seen is they have peanut allergies a lot. And that was the main restriction for us to not use peanuts. But otherwise, peanuts, they're a great protein source. They have a very diverse nutrition profile. The protein functionality in terms of, making a set yogurt is good. So definitely peanuts, they are like a much better raw material from that perspective if I see the gelling ability. So.
And another question is from Sukumar Devnath, sir. Did you ever try to get milk from leafy vegetables?
No.
Okay. Another question is from Sanika B. What do you think about mixing of vegan milk for product development?
Mixing of vegan milk for product development?
Yeah.
Okay. So mixing with dairy milk, I'm assuming, right? Or just mixing different vegan milks?
They are not clarified here. So maybe mixing of vegan milks, CS wrote.
Yeah, I think that is actually a good idea because if we do that, Mixing with dairy, I don't feel it's a very good thing, because then you cannot actually cater to the vegan market, to which like these plant-based products are actually catered to. And mixing different vegan milk options, it's a very good idea, because for instance, we know like soy milk definitely structures, it forms gels, whereas almond milk does not form form a set gel if you inoculate it and take it through conventional yogurt process. We know that these two, the behavior of these proteins are different. So if we combine them together and use certain ratios, then definitely we can we may be able to get a good set gel and a diverse nutrition profile. So mixing two things, and specifically, I feel that from a nutritional standpoint, I'm so proud of actually being an Indian. Like even before all this food science research came up, we actually knew that grains and legumes together make a complete protein profile. So probably if something can be combined in that aspect, because almonds, I think they are pretty similar to legumes in their legumes. They're pretty similar to cereals. So okay, cereals are deficient. I forgot. Cereals are deficient in lysine, or legumes are sulfur rich, and cereals are lysine rich. Please correct me if I'm wrong. And so almonds are particularly, they are like cereals. They are lysine rich, but then they don't have the sulfur-containing amino acids much. So probably if we can combine them with something like a leguminous source like soy, we can actually get a complete protein profile. So that's actually a really good idea.
Devnath sir, you can ask your question directly also.
Yeah, Dr. Bhanu.
Yes.
So excellent presentation. Specifically, your PPTs are really excellent, wonderful.
Thank you.
Yeah. Actually, we tried that long back, maybe 2012 onwards, to get some kind of green milk from the moringa leaves and something. I think sir also knows, because he was also our CSR, that RC expert, actually, something. So we tried the milks, we are able to produce it from moringa, but it was not done large scale. Actually, production was a little difficult, actually. So we stopped it, actually. About 25 persons worked on this to get it into the market. Somehow it was not materialized in a large scale production, something, due to some problems or something and all this. My question is that, so I thought you will be trying from different sources, and will be like we do the blended oils or something like that. Someone asked me just now, the blended milk and all this. That will be a very good proposition, I think. The scopes are there. People want it, actually, based on the different fat sources and the protein sources, something like that. And secondly, is there any problem for some kind of flatless problem, like soy-based milk or soy-based product we have? In case of the almond milk, so that I just want to know. Do you think, is there any, if you have almond-based milk and some product, something like yogurt, is there any problem, like flatless problem, that we observed for soy milk or soy-based product, something like that?
I don't think, so sir, for flatulence, I don't think like for almond milk, I have had almond milk myself, and I particularly don't react. So I was basically in a vegan household. So based on that experience, back in Australia, so we were like 4 girls, vegan vegetarian. So based on that experience, it's like I can say that almond milk does not cause any flatulence, because it was basically their favorite. So almond and oat were the two favorite options of everybody. So I don't think it causes flatulence, but I don't have like any specific experimental tests that I have executed on it.
Although almond milk is available, but because of very high cost, we have never purchased in the local market. It is available, but it is very high costly. It is
I know. And in fact, one thing that I would like to share here, that industrial things. So when I was back in India, I checked like all the products and everything. So I saw that the commercial samples, they would also have like less content of almond. They won't really have like a lot of almonds, but still, and that's why they will be like low, advertised as low calorie, but people, what they don't realize is like low calorie is also low nutrition. So please don't go for like low calorie stuff. So anyway, they will have like much less almonds. So if you, and they're like priced at like around 250 to 300 per liter in India, which is insane. But I think in India we also have peanuts, which is very good. They are local to us. We have good amount of peanut production in India. They're nutritionally richer, and I'm not sure, but until now, I have not seen anyone in India, not met anyone in India having peanut allergies. So probably they are like.
But I just wanted to tell, peanut allergies are there. Many people cannot take it actually. This peanut-based product and something like that. So that way it is advantageous and more nutritious also. If you, it's a good approach and all these things. But as I told that if you are able to blend it with some low-cost product, which is rich, nutritious, low-cost and nutritious product, it will still better and something. Another way I'm very happy that because Gargi Ghosal is my junior actually, so we came from the same university. So I am at CFTL and she is, and I get sometimes thesis also from that university. So I'm very happy to hear his voice and all this after a long time. So thanks to Professor Mishra sir, Guruji, pranam sir.
Thank you
very much. And thank you, excellent presentation. Very happy to see you. Thank you very much.
Thank you, sir. So, Mitra Banerjee, he has raised the question.
Hello.
Like hand, so he can.
Dr. Bhanu ma'am, really beautiful presentation. Am I audible, ma'am?
Yeah.
Yes, you are.
Ma'am, I am having one question. You have said that at pH, you have used almond protein isolate you have used, and at pH 4 and pH maximum. So, usually the isoelectric point is around 3.5 pH. So, where this protein isolates are usually prepared by alkali washing and acid precipitation method, ma'am. Where precipitation occurs at between 3.5 pH. So, was that normal protein isolate or something advanced than that, ma'am?
I'm not sure if I'm clearly understanding your question because the voice was breaking a little bit. So I'll try to answer, and if it's not good enough, please say it again. So the thing is, so I prepared the protein isolate using salt extraction dialysis method and not the AEIP method, because the AEIP, which is like the alkaline extraction method, it was not giving me a very soluble product, and we were having issues with the solubility. So I showed you here that this isolate was pretty soluble at 4. We did have for almond proteins, if you do there's a potential measurement and things like that, so you will notice that the PI is around 5 for almond protein. So, but I still, and also because it's like a diverse profile, as I said, a mandan major almond protein is just 65% of the total protein amount. So, and we can get only 65% precipitated at pH 5, and we are losing the rest 35%. And because we didn't want to lose the rest 35%, we didn't go with the AEIP method. We actually just went with the salt extraction dialysis because we thought it's good to have the entire protein. Almonds are already expensive. We don't want to lose the protein. So.
Thank you.
Another question is from A.K. Sharma ji. So, what are different stabilizers you tried and recommendations for future research on product development?
Okay, so first of all, stabilizers, I didn't use any because I didn't want to because so as I said, the industrial products, all the almond yogurts that I showed here, so they all had stabilizers in them. But what my experience has been, by adding the stabilizers, you cannot definitely replicate, as I showed in the tribology measurements and the when we measure the rheological measurements, you cannot totally replicate the dairy behavior. So you need to have that structure which actually gives that particular behavior. So that's why I'm not a big fan of using stabilizers, because I feel the less products, the better is the product. The less ingredients in the product, the better is the product. So, and if stabilizers are not giving me, so mostly the stabilizers are added for preventing syneresis or for water, just preventing syneresis, water binding, that's right. But otherwise, so that is okay, that's a good application. But otherwise, to just increase the thickness, I don't think, based on my results, I can say that it actually helps in thickness, creaminess, or smoothness. So yeah, so I'm sorry I cannot help you with this. That I am not sure about what stabilizers, because I've seen people using gums and starches, of course, for thickness, and gums to prevent the syneresis. So, but I'm not very sure about like what stabilizers to use. And the future research, of course, is so first of all, so we know that we can get these kind of interconnected protein networks from almond, but then in that case, so very good point that was pointed out by other people was mixing it with something which can increase the nutrition value, lower down the cost, and also improve the product properties. So that will be a very good thing to do, and also. Due to some specific reasons, like I could not include directly the almond yogurt formulation in my thesis, because we had an industrial collaboration, so we have to keep all that thing out. So I can't comment on that, but I would say that there is a lot of scope in improvement of the texture of almond yogurt formulation as well. So apart from that, another thing is like plant-based cheese. Definitely haven't tried that, and so I definitely want to go and try like all of these options and see how the proteins react. But yeah, I think for cheese kind of products, I feel one thing that I have realized after using almonds in my study is that it is definitely good to have like raw materials which are not very expensive and proceed with them, so that they are more affordable to the market, specifically now, because more and more people are reaching out for vegan alternatives. So this I definitely feel that should be the way ahead.
Then what was the shelf life without stabilizer?
Again, so I did not use any stabilizers. So shelf life was 5 days without stabilizers. So after that, the shelf was a lot more than stuff.
Okay. So one question is from Ajit Kumar Singh. In soyabeans, some anti-nutritional compounds are present. So how does it remove before preparing the soya powder?
Okay, so again, I don't have a lot of experience with soy, but with almonds I can comment. So because for almonds we knew that there are polyphenols which actually restrict absorption of nutrients. So that is why, again, like I love India in this manner. So we have like a lot of wisdom from our dadis and nannies, and they always say like, badam ko soak kar do raat ko, and then subah uska chilka utar ke khao, right? So that's a very good way of actually, it's basically soaking that we also use for like pulses, for like nuts as well. So that actually, when we remove that, we get the polyphenols out and it increases the nutrient absorption. So that is what they, I know about, like for usually for pulses, but for soybeans, I'm pretty sure that there will be a difference, like the nutritional, anti-nutrients will reduce after soaking, but to what extent, I cannot comment about that.
Soaking as well as heat treatment both can be done to reactivate the nutrients in soybeans.
Okay.
A lot of reports are there. Soaking as well as heat treatment both.
Okay, sir.
Yes, sir. Heat treatment will be the best one because trypsin inhibitors will be there, so that gets out actually.
Right.
Yes, sir. Okay, sir, you can ask your question directly also. Like maybe you will require a discussion instead of line by line.
This homogenization, definitely it will require to have. Yes, it will depend upon the particle size, etc. what is there, because it may settle, otherwise it may settle. It will not be a product that is uniform like milk. All right. After some time, any cereal, that is the problem with any cereal or grain beverage. Do you agree with
problem?
Bhanu?
Yes, sir.
Do you agree with me what I told, or
Yes,
I
told.
what is the product required homogenization? But
yes, definitely.
if you homogenize it, then also it will settle after some time. But that you will have to manage with the particle size, like micronization. And if you
Yes,
the particle size, okay, then only you can have particle size reduction and homogenization and then proper packaging. Still, the problem difficult in grain beverages.
Yeah.
Before consumption, you have to shake it and then mix it and then use.
Same is the case with almonds also, sir. Same issue.
Any grain beverage, this will
Yeah.
and that is always, although in the terminology they are using this almond milk, peanut milk, and all those things, but that is actually not a correct terminology to use it. Milk basically is the lactal secretion. So you can say that is one problem that what Dr. Devnath was trying to say. कि almond milk तो इतना महंगा होता है हम नहीं खरीदते हैं तो क्यों जब मिल्क 12 रुपया लीटर हमको यहाँ या 40 रुपया लीटर 50 रुपया लीटर अभी मिल रहा है तो 300 रुपया लीटर almond milk क्यों खरीदेगा in country like But if you introduce it as a replacement of a beverage, cold beverages and all those things, nutritional beverage, and then compare the cost from the Coca-Cola bottle, 200 ml Coca-Cola bottle, you are paying 20 rupees.
Yeah.
Right? So,
right, sir.
And without any much nutritional value.
Right.
So, if beverage this, compare this milk with that sort of beverages and introduce in the market, it may have some.
Fine.
So,
exactly. But everyone cannot buy it. Everyone cannot buy it.
Cost is a very big limiting factor.
These are luxurious products.
I have seen
milk is available in plenty. So people will buy milk. Why they will go for grain milk and alternative milk? Until they are having some specific problem like lactose intolerance.
Exactly.
True.
For them only.
Nice presentation and also nice discussion.
Thank you.
Sarma ji, आप थोड़ा सा you can comment. Yes, cost depends on solids. Definitely, it will.
Yeah.
So Shubham.
Yes, sir. So any participants having any other questions or queries?
No, sir, wonderful presentation. Today I am not seeing Dr. Vishwakarma.
Yes, sir. He joined it actually.
Actually, Vishwakarma, he has gone. He is having some engagement in Kolkata.
Yes, sir. So he went to Kolkata. That's why.
Yes.
Okay. Hi, Gargi. Just a glimpse. Gargi, I have to talk to you after this. After long back, I am seeing you. So many years. Okay. Congratulations to you also. And to your former student. Thank you, sir. Thank you, Sirji. Thank you very much.
Thank you.
I have a question in the discussion now. What is your opinion about oat milk alternatives?
So they are good. It just depends on your taste preferences. So with the plant-based stuff, taste matters a lot. So what I have realized again on the basis of my experience of living with vegan people, that they feel that the taste and the flavor of oat milk is very neutral and close to dairy. Although, to be honest, I tried it and I'm a lover of dairy. It was very hard for me. And I just get back to dairy. I tried almond, get back to dairy. Try oat, get back to dairy. So, but yeah, like from nutritional standpoint, it just depends. It's like your personal preference. Like if you like the taste, if you like the consistency, so oat products to prepare to tend to get that creamy consistency in them, which is good. So yeah, I think it just depends on your personal preference, like what nutrition you would like to have. If you want, to get protein and calcium from the milk, then oat comes into question. So it just depends on your personal preferences. But yeah, it's just every different nutritional profile, so whatever you like.
Yeah, nutritionally it is better, but oat milk is little difficult to prepare. That is it. Almond milk is easier to prepare. Peanut milk is easier to prepare. But oat milk, you have the starch, solubilizing the starch and getting it emulsion like milk is little difficult, but not impossible.
Yeah. So there are already alternatives available, I'm pretty sure that India also, I think that the Good Milk, the company, I think now they have changed the name. I'm not very sure what's the name now, but they do have like cashew oat milk. So they have like a blend of cashew and oats. So yeah, so there is, it's like in India, we do have, I think, oat milk in the market commercially.
Yes, yes.
Yes, sir. What milk is here?
If you could go less starch-based grains and convert it into beverage, it may be a good idea. Otherwise, the starch, like yes, millet is also millet milk, etc. But again, the starch will have a problem. Good.
I think Bhanu ma'am left.
Huh?
By mistake, she left.
She has left, huh?
Yes, sir. By mistake, she left, I think.
And maybe some network problem.
Yeah.
Anyway, you can continue and you can do the customary. Thanks.
Yes, sir.
Sorry, sir, there was a problem with my connection again. So now I'm just like through my laptop, so audible, but not, you can't see me.
That's fine. That's
Sorry
fine.
about that.
Good. That's fine.
One question is from Sunny. Millet milk is more cost effective than almond milk. What do you think?
Yes, that will be, no doubt about it. So again, that's the good part about India, that we actually have a lot of diversity in terms of pulses, in terms of grains as well, you know. So that's a really good thing. We have like our own millet production in India and good amount. So yeah, definitely it will be more cost effective. But I think you'll again face the same challenges as sir mentioned with all the grain beverages. When you have more starch, you'll have to definitely give it some enzymatic treatment to break it down to so that it's stable.
Richard has raised his hand, so you can ask Richard.
Sir, good evening, sir. Sir, sorry, actually I missed today's webinar, sir. I had up some traffic actually. I could not attend, sir, actually. So, can we get it this thing, sir, video, this one, sir, today's webinar?
Yes, it is already, yes. You can send the link, put the link on the...
All the videos are available on the Facebook page of FCTL IIT Kharagpur, or you can go to YouTube of FCTL IIT Kharagpur. You will find the videos.
Okay, fine. Thanks a lot, sir. Thanks.
Of this webinar as well as the previous all the 44 webinars.
Sure, sir. Thanks a lot, sir. Thanks.
Yeah.
Thanks.
Okay. Okay, ma'am. Like.
you can now conclude.
Yeah. So now we can conclude it. So I will invite Priyanka.
Thank you, sir. Good evening to Dr. Bhanu Devnani, Mishra sir, and my dear seniors. It is my great pleasure to put forward a vote of thanks for this 45th lecture of FCTL webinar series. I warmly thank our speaker, Dr. Bhanu Devnani, for sharing extensive knowledge on structuring plant-based dairy alternatives, its challenges and opportunity. Ma'am, you made us aware why plant-based dairy alternative came in place. Then two approaches of getting plant-based dairy. First one is minimally processed system, and the second one is the purified system. Then compared almond, soy, dairy yogurt, saying its microscopic structure, viscosity, friction coefficient, etc. You taught how protein that leads to aggregation and gel formation in almonds. At last, we saw protein-based network structure could be obtained with almond protein with all the curves. Once again, thank you, ma'am. I am deeply grateful to our mentor and coordinator, Professor N Mishra sir, who has given us the opportunity to gather knowledge via this webinar series. Lastly, I would like to thank the audience and to all the seniors who took time to impersonally participate in this webinar series. Thank you all. Over to you, sir. Thank you.
Yeah. Chirasmita ma'am bhi aa gayi hai.
Yes, now she can interact. Yes. Bye-bye.
Yes, sir.
Good. The seminar is formally over. All right. Thank you all. Bye-bye.
Yeah. Thank you all for participating.
Thank you. Thank you so much, everyone.
Next seminar will be on 29th October.
It's good. And the
Thank you, sir. Thank you.
Will be given. Yes. Good. Thank you. Bye-bye.
Thank you. Bye-bye.
Thank you, sir. Thank you very much.
Okay, ji. Bye-bye.
Bye.
ठीक है भानु, stay blessed. All right, thank you.
Thank you, Bhanu ma'am, for the great lecture.